Thank you, Michelle, and thanks for the opportunity to have this webinar and with the three very interesting presentations and an important topic today. And I've been giving a few minutes and have a very high level and looking at the unmet clinical needs in predicting responses in oncology drugs, and then ultimately say how AI can help, which the three speakers is going to guide us through. And in a very, very high level and 10,000 foot look at what is unmet needs in drug discoveries. And we have the number of many, many challenges. But if you look at the categorical challenges, and the first would be in the biomarker identifications, and it's really the limited biomarkers we have, and the many cancers are lack of reliable biomarkers. And also the tumor heterogeneity complicated this situation. And the genomic and the molecular profiling, it's another area that we've been continuously working towards. And the drug resistance, clinical trial design. And so all the things, and now actually AI is holding a good promise to help. And some of our presentations today is going to demonstrate that what is the current work and the future directions can go with that. But furthermore, if you look at it of the drug discovery, and you will see that the patient-specific factors is also one of the challenges. Maybe AI can play a big role there. It's actually looking at a personalized medicine, tailored treatment to individual patients there based on their unique genetic markups and tumor characteristics, and their immune system factors, how to understand how their own immune systems are going to respond and impact the drug responses. And the data integration sharing, without saying, we still have lots of challenges in this area, and ethical regular considerations. That's all reflect on that the challenges we have, and AI may actually help in alleviating some of the restrictions and the cost accessibilities, without saying that it's always a struggle, and hopefully that we can have some help from the advanced algorithms and the better data sharing agreements and accelerated drug discovery. So we'll talk about AI, and I can get another 10,000 foot of what is AI. And other people will say AI is a science of intelligence, etc. But the latest definition is that an AI system is a machine-based system that for explicit or implicit objective infer from the input it received and generating the output, such as predictions, content, and the recommendations or decisions that can influence the physical and the virtual reality. So that is actually the definition, the latest definition of AI. But if you look at hierarchical terms, and in the past, we kind of sometimes interchangeably using the machine learning and the AI and the deep learning somehow. So now the whole hierarchical structure of in this domain would be you have the larger, more general terms of the artificial intelligence as we described before, and or even further, the universe of the data science. But then the machine learning, the certain algorithm itself, and is considered as a subset of the artificial intelligence. And the deep learning, we talk about the CNN, RNN, and some of the topics, algorithm mentioning in the following presentation today, and would be considered as a deep learning as another subset of the algorithm itself. But then you can see that there's some other dimensions you can put it in. And one of the things could be the reinforced learning and model-based control and the decision-making under the uncertainty. So that is quite relevant and quite prominent in the application in the healthcare and in the research work we are doing here. And you can further identify that some of the specific topics in AI, such as natural language processing, imaging processing, biomarker, and genomic sequencing, the AI algorithms can cut across the machine learning, deep learning, and so then there's a slew of the other dimensions you can have. And the biomedical, bioinformatics field is kind of a specific fields can cut across all these actually domain as well. So if you look at a very specific another way of talking about the whole metaverse of the machine learning and AI. You can see we have the supervised learning, semi-supervised learning, enforced learning, and unsupervised learning. That's each of the categories of the algorithms can actually apply for the solving the different purposes, either for discovery or for classification, diagnosis, prognosis. So if you look at this, actually algorithms is a whole thing about all these words mentioning here, representing one types of the machine learning algorithms, and you can have, and I'm not going to go through the detail, but you can see that there's lots of actually algorithms, variations of those algorithms at our hand and in the arsenal that we can, as a scientist, can explore. Okay, so if you look at the very short history of the AI and machine learning, it's not new. And in the 1980s and even beyond that, we actually started to injecting the human knowledge, expert knowledge, and learning something into learning from the data, learning from actually the experiences of the human, and convert the machine, our knowledge into the machine processable form and help us to make a decision. That is actually nowadays, we all call them AI, but actually in the old days, we can call them the decision support, and we can call them expert system. We can actually call them also the computer assisted diagnosis or detection CAD. That is in the imaging processing part of it was being long term, and for last 20 years we talk about CAD, and now we're all talking about the AI. So you can see that there's a long development, and up to today, we are very heavily focused on the deep learning, and the generative AI hold some great promises. And the computing power is significantly increased in the last decade, and the cost of storaging and also computing is dropping down. It's not like we say we actually do not think the AI will be very expensive. The research actually presented following would have been very expensive, but still that we have much, much more powerful tools and the computing infrastructure to deal with a large data. So let's see how they can actually help us solving those unmet clinical needs. So other results that we're going to introduce, is my pleasure to introduce our first speaker, Dr. Laurent Huffer. He's a research scientist at the Drug Discovery Group as OICR. And so I'm going to saying that Dr. Laurent Huffer is his focus is on the computer aided development of molecular probe and potential potent inhibitors for diverse targets, including broad domains and the protein kinexes and the PDZ or WDR domain containing proteins. And Dr. Huffer received his PhD in chemoinformatics from University of Strasbourg, where he specializes in molecular docking of the small organic compound and their optimization through in silico methods. He pursued his postdoc at the Cancer Research Center of the Marseille, and where he developed the molecular modeling methods and dots and the cover dots for structure guided optimization of the compound. So it's my great pleasure to have Dr. Laurent Huffer for his presentation.
So good afternoon, everyone, and thank you again, Hélène, for the nice introduction. So first, I'd like to thank the organizer for his invitation to this nice OICR Monday event. And it's a great opportunity to talk about the application of artificial intelligence to the drug discovery process. So basically, I wanted to talk about AI in a nutshell, but Hélène already mentioned a lot of things. So I will just go very quickly through the first slide. So today, we'll talk about AI and machine learning. In practice, I will mainly talk about machine learning, but most people will employ the expression AI, ML. So the main stages can be described very quickly. So first, a model can be built starting from input data using some training or learning algorithm. And then the model will be challenged with some statistical validation to evaluate its robustness. And if the model is good enough, we can move to the prediction. So even if AI ML is pretty new and fuzzy for the general audience, most people already use it through mainstream applications such as ChatGPT or just the creation of images from prompts. You'll agree with me, this is a pretty nice cat picture, but what about AI in sciences? So the same logic basically will apply in sciences. However, various types of input data can be used, leading to different types of predictions. And as highlighted by the three different talks from today. So in the drug discovery field, we'll mainly use sequences, chemical structure, or descriptors encoding these molecules. And I will mainly talk in my talk about all of these highlighted properties prediction in green. So next, Cathy Scott from Adaptive Oncology Department will discuss the use of medical radiographic image to predict some drug response. Finally, on design, from clinical department, translation department, will also discuss the drug response, but using some completely different input data, such as multiomics. So what is a drug discovery process in a nutshell? So this chevron here represents the modern drug discovery process, and it starts with target identification. So then some lead compound exhibiting some activity against the target of interest are identified, starting from a large collection of compounds. Then the hits are improved, for instance, a better potency to the target, to become optimized compound called leads. Then the leads are improved to produce a potential clinical candidate suitable for clinical trials. So up to this point is so-called drug discovery. So next, we have drug development that will include clinical trials, but this is beyond the scope of my talk. So now, where can AI be involved in the drug discovery process? So I will focus on the few compound development stages for the purpose of this talk. And long story short, AI can be applied to every single stage with diverse methods. So due to time constraints, I selected three main categories, and I will showcase one famous example for each category. So then, another main promise of AI/ML is the ability to better predict some binding energies. So basically comparable in terms of accuracy here, comparable to in accuracy to quantum mechanical level, but much faster. And in parallel, some very large, huge chemical databases were created, mainly by chemical suppliers. And AI/ML methods are particularly useful to navigate through this gigantic library. And I will make a link with the virtual screening. And I will also discuss a few additional AI applications of OICR drug discovery at the end of my talk. So let's start with AlphaFold. So AlphaFold is a deep learning technology that was developed by DeepMind, and its goal is to predict the 3D structure of protein starting from their sequence, 1D sequence. So the original paper has been already cited by 24 times. It's only from 2021, so highlighting basically the huge impact of this paper. So by contrast to experimental method, in AlphaFold, all the amino acids from the sequence will be modeled with diverse confidence level here. And also, AlphaFold achieved some groundbreaking performance in the Cas14 protein folding competition by a large margin. So AlphaFold has a potential huge impact in both structural biology and drug discovery. And all the predictive 3D models are available. You can download them. You just need to go to the website. And finally, the main competitor from AlphaFold is RosettaFold. So Now, I will just cover the main uses of AlphaFold models. So first, we can use a model for general structural analysis. And these models are great resources, particularly in absence of any experimental structure for the protein of interest. Then, these models also have some valuable hypotheses for the refinement stage in structural biology, especially in cryo-EM. However, this AlphaFold will not replace experimental structure. And finally, these models can be used in molecular docking or structure-based virtual screening. I also, and basically we use AlphaFold model almost on a daily basis at OICR drug discovery. So I also need to mention that AlphaFold 3 and Rosetta Fold, so the new version were just released just a few weeks ago. And now they have the ability to predict full protein ligand complexes. So previous study highlighted some pretty important limitation of AlphaFold 2 model for use in drug discovery. So now the question is, with AlphaFold 3, so will AlphaFold 3 lead to groundbreaking impact in drug discovery? Time will tell. So let's discuss another use of AI in the context of virtual screening. So basically, very short, the goal of a virtual screening is to identify compound from computer simulation, for instance, molecular docking, and where large library of compounds are considered to prioritize a small number of compounds. And some people from UCSF conducted a few years ago, and it's a paper from Nature, some very large docking studies, and they tested systematically compounds from different score bins. And they basically demonstrated, as expected, there's a correlation between the hit rate and the docking score. So obviously, you will still find inactive compound with a great docking score, but basically the hit rate will be much higher than with a low score here. So if you combine this with the fact that now we have access to very large library, we should be able to prioritize this high docking score by screening virtually gigantic library. However, the systematic virtual screening of billion compound is still not possible, even if you have access to huge computing resources. And to bypass this limitation, some alternative methods were developed. And long story short, basically in the middle of the process, a machine learning model will be trained to predict some docking score starting from the structure of the ligand. And then we can use this machine learning model to profile the initial gigantic data set to identify the most promising compound according to the model. And only this best compound will be explicitly docked. So long story short, the goal is to concentrate the computing resources on the most promising compound, where very good docking score is expected, and where we can expect also higher hit rates. So this method has been adopted for recent and future virtual screening at OICR. So we just took a brief look at some structure-based method where AI can be employed. So these approaches are great to find hits and to improve potency. However, drug discovery is much more complex than that. And we need to improve a lot of different additional properties. And these properties are usually competing between each other. And when you improve one, it will be done to the detriment of another one. So usually multiple experimental rounds are required to optimize the compound and then to match the target product profile. And the target product profile just lists the different endpoints, properties, with some basically objective values. So ligand-based methods are well suited for multi-parameter optimization purposes and very helpful in the drug discovery context. So imagine now, if we have some predictive AI/ML model for each endpoint of interest, we should be able now to perform some virtual rounds to try to improve the compounds. And on the right, you can see the typical ligand-based workflow for this kind of virtual design workflow. So this one is called GTD from BioVia. I do not have time to describe the process in detail, but the take-home message is if you have access to some automated design and some AI ML model for every single endpoint, you should be able to prioritize compound matching the desired target product profile. Obviously, to do this, you need a lot of high-quality data. And this technology is used in both academia and companies. So as mentioned earlier, so we regularly use AlphaFold models and active learning docking in our project. So now let's discuss some additional AI application at OICR drug discovery. And starting with the partnership we made with BioVia. So basically, so again, GTD is a cloud AI ML drug design platform able to perform the virtual design strategies that I described quickly on the previous slide. And we applied this approach on a very advanced protein kinase project involved in the hippo signaling pathway. And basically, protein kinase inhibitors can be new potential therapeutic for cancer. So we had some issue to optimize all the properties at the same time, as in typical drug discovery development. And we, thanks to the chemist and biochemist, we have access to high quality experimental data. So we can train some machine learning model for every single endpoint and then use the platform to propose, to suggest new designs. So now we'll quickly describe some experimental results we got without disclosing any structure. So, and we also in a lead optimization context. So the medicinal chemists from our team did a great job and generated a lot of good potent compound and even from different sub-series. So we mainly investigated one series, and we can see that the starting lead is very potent on the target, very selective against 3 anti-target. Only one endpoint was problematic, the stability in mice. So, but this was a no-go. So the medicinal chemist selected the most attractive compound from a pool of design compounds, and then about 20 compounds were considered for testing. So as expected, different profiles were obtained. So sometimes the compound was not potent enough. Sometimes the compound was not selective enough. Sometimes the compound had the same liability for the ADME properties. So not all the predictions were correct. However, we still got some very clean design matching the complex TPP and improving some known issues. So obviously, the compound still requires optimization to evolve into a potential clinical candidate. And I would conclude by there is no black AI/ML magic. So you don't expect this kind of method to design from scratch some clinical candidate first try. The success will likely be a combination between the human experience from the chemist and the use of AI here and there. So now I will finish with one of the latest endeavors, namely QXL, which is a joint venture between OICR and Cogniframe company. So its philosophy is pretty simple. It's to leverage AI/ML approaches for efficient drug discovery. So previously I mentioned that AI/ML model have some ability to better predict binding energy, so comparable to DFT or QM level, but at a force field cost. And this is very interesting characteristic from AI/ML. So then the technology has been fine-tuned for protein ligand complexes, and different software solutions for structure-based drug design of ligands were developed while allowing an efficient exploration of the chemical space. This QXL technology has been retrospectively validated on OICR data, and we just started some prospective use in your project. And Dr. Hugo Borges, who is a senior research scientist in the team, will present soon a dedicated talk about QXL. Let's go to the conclusion. So AI/ML achieved some significant breakthrough over the past decade. We have seen a lot of potential application in drug discovery, Long story short, all stages can benefit from AML, and a lot of different approaches can be employed. It is pretty clear that AML will play a bigger role in future. And finally, the journey doesn't end once a drug hits the market, because the patient response can vary. And this is another case where AI can play a significant role, and Kathy and Andy will discuss this in their talks. So this is a picture from our great drug discovery group. And thank you for attention, and feel free to ask any questions in the chat later. Thank you.
Yeah, thank you, Lauren. So our next speaker is Ms. Katie Scott, and Katie's received her bachelor and a master's degree in Queen's University, and her studies specialized in computing, in the biomedical computing, and also artificial intelligence. Katie is a research analyst in the bioinformatics and the computational genomic lab at the UHN. Her research focused on applying the computational methods and the machine learning to extract the information from the radiological image, particularly CT, MRI, and explore how imaging features can be associated with clinical, genomical, and other data types to improve predictions of patient outcomes and therapeutic responses.
Thank you so much for that introduction. So like Helene said, my name's Katie Scott. I'm a research analyst in the BHK lab at UHN. And today I'm going to be talking about using radiology imaging data to predict drug response. So to fully understand radiomics, we first have to establish where the information in images comes from. So in a black and white image like this, we have pixels that are different shades of gray. And the intensity or gray level of that pixel is represented as a value between 0 and 255. So when we're processing the image, all that's being passed in is an array or a matrix of numbers. And this allows us to perform mathematical transformations and extract patterns from the values. In medical imaging, the idea is much the same. Those numerical values become a little more nuanced as Hounsfield units in a CT, which have a larger numerical range, but the underlying idea is the same. If we looked at a tiny portion of this slice of a CT scan of the lungs, we could extract the values from it and perform processing on those values. In imaging analysis, we typically want to focus on a single part of the image, such as a lesion or tumor, so we can create what is known as a segmentation or a contour. This segmentation let us look at just the pixels that are included in the region of interest, so they're highlighted in green on this image. And the way that we represent this is with a binary version of the image. This will have the same dimensions as the CT or MRI, and pixels we're interested in looking at will be marked with a non-zero value, such as 1 in this example, and everything in the background will be 0. So with the CT and the segmentation combined, we can perform radiomic analysis. Radiomics aims to extract underlying biological information from medical images that might not be perceivable by a human being. This can be done on a CT like the one shown here. So first, that region of interest, such as a primary tumor, must be contoured so that we can identify where to extract information from. The voxel values or pixel values within that contour are then used to calculate radiomic features that are related to intensity levels, shape, and texture of the region. There have been multiple open source toolkits developed, such as PyRadiomics, LifeX, SIR, and Captic, but none of these are consistently used across the field. Radiomic features are most often shared either as a supplemental part of an imaging dataset or in the form of a radiomic signature. This signature is made up of one or more radiomic features in combination that are shown to be capable of predicting a clinically relevant outcome. One issue here is that there's no standard method to share these data sets or signatures, and that's even if they're made publicly available. And this hinders the possibility of external validation, which is a prominent limitation in not only radiomic studies, but in most AI studies. Radiomic features have been shown to be helpful in risk stratification, detection, staging, and treatment planning in various cancer types. And for today, we'll be focusing on treatment planning with regards to drug discovery and response prediction. This work by Roger Sun et al. Is one of the most prominent successes in the field of radiomics and explores response to a specific type of immunotherapy. In this study, radiomics analysis was applied retrospectively to 4 independent cohorts of patients who had solid tumors from various cancer types. The first data set was used to develop a radiomic signature to determine the quantity of CD8 cells, which is a measure of immune response. This signature was then validated in the 3 remaining cohorts to explore association with CD8 cell gene expression, tumor immune phenotype, and clinical response to anti-PD-1 or anti-PD-L1 immunotherapy treatment. There were 8 variables that made up this signature. 5 of those were radiomic features extracted from CT images, 2 related to the tumor's location, and 1 pertaining to the CT scanner's max voltage. This radiomic score provided a promising way to predict the immune phenotype of tumors and to infer clinical outcomes for patients with cancer who had been treated with anti-PD-1 and anti-PD-L1. The other reason that this paper is highly regarded is that this signature went on to be validated 5 times in additional works. 3 of these are from the original authors and 2 are from external groups. One other significant work from radiomics I'd like to highlight is this paper by Aerts et al. This work showcases one of the first radiomic signatures that was developed on lung and head and neck cancer data sets. Their signature was developed on the lung cancer data and included 4 imaging features from intensity, shape, texture, and wavelet texture categories, respectively. This signature was then tested on a separate validation lung data set and 2 head and neck data sets. The predictive models built on the signature all performed well, particularly in the head and neck model. This work went on to be expanded into one of the most popular radiomic feature extraction tools called PyRadiomics. Now, something to note about this work and other works in the radiomics field is that the authors claim that these models outperformed tumor volume alone in terms of prediction. And while they may be correct, there isn't usually analysis included beyond checking the correlation of the features in the radiomic signature to tumor volume. And so some previous work from the lab I'm a part of actually explored that. In this work by Welch et al., they analyzed the same radiomic signature and data set from the previous slide and compared how predictive it was to the volume of the tumor by itself. This exploration took the images and randomly shuffled all of the pixels, but maintained the same original 3D contour. So the shape features are exactly the same, but the other classes of radiomic features should change and the model should no longer be predictive. This was not the case. As seen here, the graph D shows the model results for the original image, E shows the results for the image with randomized voxels, and F shows the results for tumor volume alone. All of these models achieved the same predictive performance. So we're interested in figuring out how to isolate radiomic features that aren't just dependent on volume to see if there is actually predictive ability that can be used in AI models. Along with this potential volume dependence, radiomics studies are hindered by a lack of standards for processing and data sharing. And like was mentioned before, having large amounts of data and being able to share them is really important for AI model development. And this brings me to the proposed pipeline that I've been working on that combines existing tools from my lab with a new package that I developed where we aim to address these limitations. And so there are 3 major tools in the pipeline: MedImage tools, Ready, and Orchestra. This pipeline was mainly developed using the publicly available RADCure dataset, which consists of head and neck cancer images. And so we start by feeding the CT and segmentation images for a single patient into MedImage tools. This tool is responsible for pre-processing the image files and properly associating each CT with its corresponding segmentation, and then passing those to the next step. These are fed into Ready, which stands for Radiomic Extraction and Analysis for DICOM images. DICOM is just the standard file type for a medical image. And this tool uses the PyRadiomics library that I spoke of earlier, but as of the time of this presentation, they don't have processing set up specifically for DICOM images, so this is what we developed. Ready takes in the CT and segmentation and performs quality checks to ensure that the images have matching dimensions and alignment. And this is where we introduce what's known as a negative control. Just before feature extraction is performed, we take a copy of the image and generate one of 9 potential negative controls. These 9 negative controls are made up of a combination of 3 regions and 3 classes. The 3 regions are the entire image, within the contoured region of interest, which is the primary tumor in this case shown in green, or all of the voxels outside of the region of interest. So if we think back to the mask, anything that was marked as a 0 would be what would be changed. So shuffle reassigns voxels randomly within the specified region. Random generates and assigns a value within the original voxel range in the specified region, and random from original distribution randomly selects values with replacement from within the specified region. Once the specified negative control has been created, the image is cropped to contain the region of interest, and radiomic feature extraction is performed. These resulting features can then be applied to analysis, such as in an AI model, and then are fed to the final step of the pipeline. This step prepares the data to be uploaded to Orchestra, which is a web app created by the BHK lab that hosts processed omics data. The standard for data organization is a multi-assay experiment R object, and this is designed to harmonize multiple experimental assays from an overlapping patient set. We also include clinical data as well as the radiomic features from the original image and each of the created negative controls, and this is uploaded to the web app so that it can be shared widely and used along with the publication with any code that was used to generate the data. And so all in all, this pipeline we've developed is known as the radiomic extraction and analysis for DICOM images to refine objective quality control pipeline, or if that's too much of a mouthful, we just call it Ready to Rock. We showcased Ready to Rock's capabilities by processing and analyzing RadCure to compare 2 previously published radiomics models and 2 trained from scratch models using the resulting data. To demonstrate the performance of the negative controls, we performed feature extraction with the shuffle and random options applied to the full image mask, which can be seen on the left of this slide. For comparison, we fit a Cox model to the RadCure training cohort using the same radiomic signature and applied it to a test cohort, and a univariable model for mesh volume was also tested as a baseline. All of these models were compared using the concordance index. This table shows the results for each of those survival models on the test set, and you'll notice that model performance is similar whether the features were extracted from the original CT or the negative control samples, suggesting that this radiomic signature is correlated with tumor volume, or at least invariant to the underlying imaging data. This is confirmed by the performance of the univariable volume model. While this first experiment was focused on predicting overall survival, we are now in the process of applying Ready to Rock to predict drug response in some clinical trials. In addition, we're also working on applying the Ready to Rock pipeline to 5 additional open source head and neck cancer data sets, and then I'll be using those to test the robustness of some AI models that were also published by my lab a couple years ago, specifically with our negative controls. One last thing I wanted to talk to you about quickly in terms of imaging for AI and drug response is to talk about where we're headed in imaging research. And so I don't know if I can't recall if this was mentioned right at the top, but one of the latest hot topics in artificial intelligence is this foundational model, which is a deep learning model like a neural network that's been trained on a very, very large data set. I'm talking millions of images or millions of any type of data type. And like I said before, large imaging data sets are not easy to come by, but now with this push in AI research, we're starting to see bigger and bigger collections of them. And so this paper I'm showing here was published by the same group that made PyRadiomics, which is the tool I used in my pipeline. And the model that they present was trained on over 11,000 images. And they showed this model parts of the image with lesions as well as parts without lesions. So kind of another type of negative control. And they showed that this model was pretty good at a couple different tasks, like finding biomarkers for diagnosis and prognosis. And so a probable future analysis will be looking at drug response. This paper was published just this year. And we're currently working on pushing our own negative controls through this model to further test that robustness. This model, interestingly enough, was published by Microsoft just last week and looks to be pretty promising for imaging analysis. And so their model works by taking in an image and a text prompt from a user, like this one asking to find the tumor core in the image. And it then segments the tumor and responds to the user with a text response as well. And it even takes, I'll play that again so you can watch one more time, takes an update suggested by the user and updates the segmentation accordingly. And so this model is really a foundational model, as it was trained on a data set of 3.4 million image segmentation and label sets that come from 9 different image modalities and 25 anatomic sites. And while the main task for this model is automatic segmentation, that is a crucial preliminary step for lots of imaging analysis, including radiomic feature extraction. So it's not difficult to imagine radiomic feature extraction becoming the next step, going in and saying, can you extract the shape features from this tumor for me? And this was really interesting. You can go and look it up. The website they have set up for this is really interesting to look at for a bunch of different imaging types, not just CT and MRI. And so lastly, just the conclusions. So radiomic analysis, like I said, aims to extract underlying info from medical imaging. And my work has provided an integrated quality control technique to test for volume invariance. And the foundational models that I showed right at the end show a real promise for image analysis. We do still need to be testing the robustness of things like negative controls to make sure that they too aren't just learning how big a tumor is as their most predictive value. And I'm very sure that applications to drug response prediction will likely be soon in the foundational model field. Just want to say thank you to all of those that helped me with this work, including my lovely lab members you can see in this picture. And thank you for your attention. And I look forward to discussing further at the end of this talk, at the end of this session.
Thank you, Katie. And I like the ready to roll. That's the ready to rock. That's great.
Thank you.
So our next speaker is Andy Zhang, and Andy is a PhD student in Dr. John Dick's lab, and he holds a CHR Vernier Award. Yay, Andy, you're a star. Okay, so Andy's research involved the application of cutting-edge technology such as single-cell genomics towards answering long-standing biological and clinical questions acute leukemia. And Andy completed a bachelor degree in molecular biology in Simon Fraser University, and now it's a PhD University of Toronto. Andy.
Okay. Thank you so much, Helen. So hi, everyone. I'm Andy, and I'm really excited to be telling you about AI and its applications in a very deadly type of blood cancer. In the lab, we study something called acute myeloid leukemia. This is a very aggressive blood cancer with very bad outcomes. Here is a little cartoon showing you that after diagnosis with AML, only about 22% of individuals will survive up to 5 years. For those who are diagnosed after the age of 65, the outcomes are even poorer, with 4% surviving past 5 years after their diagnosis. So one of the reasons this is such a bad disease is because it's so heterogeneous. And so in fact, AML is actually kind of a collection of multiple disease entities given the same label. What I'm showing you here on the left are a list of genetic mutations that are present in patients with AML, and purple means that patient, which is each column, has the mutation. It's very complicated, but I want to show that there's a lot of mutations and a lot of different patterns across these patients. And in fact, in the clinic, there are at least 12 subtypes of AML recognized by the World Health Organization and 18 subtypes by another classification. So this is based on genetics. But if we look at features such as gene expression, like how much RNA and what types are in each patient's disease, or DNA methylation, so epigenetic marks on the DNA, we also see profound heterogeneity. And each of these ways of looking at the disease can split patients up into different configurations. And so right now, there's a challenge in integrating all this information together. So on the bright side, there's been decades of research into the biology of leukemia cells and how they differ from normal cells. And that's resulted in many targets hitting various mechanisms of the leukemia cell. And this, in turn, has resulted in many clinical trials taking form over the last 20 years. There's been about 400 drugs in phase 2 clinical trials. And you can see that over time, there's been more and more and more drugs entering clinical trials, and of course, more that have been succeeding. So we're really at a pivotal point in the treatment of AML because there's all of these drugs emerging targeting different mechanisms of disease biology, and there's a question of how to determine who's going to respond to what drug. So let's take an example of a clinical trial with this purple drug over here, right? One of the challenges is that not all the patients respond, and in fact, there's some that benefit and a lot that may not benefit. And it's really hard right now to predict who are the ones who would benefit up front versus those who wouldn't respond. If you take another drug applied to the same pool of patients, you would get a completely different spectrum of people responding versus not responding. And this is something that we need to try to figure out so that we can try to ensure that our clinical trials are more successful in the future. As an alternative way of looking at it, if we imagine that Susan, a 67-year-old with newly diagnosed AML, comes into your clinic, we have to think about what drug that Susan would benefit from the most. And so how can we know in advance that it's drug C that will really prolong Susan's survival rather than any of these other drugs? So that was really the challenge that we went into. And there's many groups looking at genomics in terms of predicting response, but we took a slightly different angle to tackle this problem. Something that is really interesting about AML, and this is true of many other cancers as well, is that there are different types of cancer cells that are present within the same patient's disease. In AML, they're arranged in a bit of a hierarchy where you have stem cells creating progenitors that then create these mature cells that resemble parts of our immune system, but are in fact leukemic. So we developed an approach to quantify the abundance of these different types of leukemia cells within individual patients. We took RNA from individual leukemia cells labeled as each of these categories and went into bulk RNA samples from over 1,000 patients with AML. Using this machine learning approach called CyberSort, we were able to deconvolute these patient samples and get the abundance of each cell within each patient. So if you can imagine, we're kind of getting the recipe of each patient's leukemia. And we were able to use that data to re-stratify patients into biologically and clinically meaningful groups. So here's a group of patients that we identified where there's a preponderance of these stem cells. And compared to all the other patients, they have worse survival outcomes. Another group of patients have an abundance, a dominance of these progenitors, which tend to be quite sensitive to chemotherapy. These are patients that have favorable outcomes. And then we have a third group of patients that have a lot of these differentiated immune-like cells. And these patients were kind of in the middle of the road in terms of outcome. And so this showed that our approach was clinically relevant, but where we were really interested was the drug sensitivity data that I'll show you now. So we went into these primary AML samples. We were able to get the abundance of each of these cell populations within their tumor. And for many of these samples, about 200 of them, these patient samples were put onto a dish and screened for over 100 drugs. And so we had drug sensitivity data for each of these patient samples that we could then relate to the abundance of each of the cell types in their leukemia. So indirectly, we could get a sense of which cells were sensitive to which drugs. So here's a map of those results. You can see on the rows, we have the different types of leukemia cells that are present within a tumor. And in the columns, we have the different types of drugs that were screened for in this experiment. And red means that it's sensitive to the drug, and blue means that it's resistant. And you don't need to get into the specific drugs, but what we saw was that the different types of leukemia cells that could coexist within a patient's tumor, they had different drug sensitivity profiles. And so this was something that was really important for understanding and predicting drug response in the clinic. So I've now described to you all of these different ways of looking at leukemia, right? Looking at the gene expression, at DNA methylation, at cell composition. And all of them are really biologically meaningful. But the issue is they might provide information that other modalities don't. So how can we integrate all these data sources together? So this was the next challenge that we had tackled. For this, we were inspired by a method from Bo Wang's group in Toronto, where they derived patient similarity networks from each of these modes of data. So each of these dots is one patient, and they're connected with each other based on similarity in the expression patterns of their RNA. And here it's the same patients as the dots, but now they're connected based on similarity of methylation patterns in their DNA. And you can see that RNA connects some patients while the DNA connects other patients together. And there's information that is complementary from these two types of data. So Bo Wang in 2014 described a way to fuse that data together and to kind of get the best of both worlds, to come up with an integrated network that used both DNA and RNA information to combine patient samples based on their similarity to one another. So this was called similarity network fusion. And we were really excited to try this on our own data, but we ran into one problem. And that was that we didn't have the same number of samples for each mode of data. So we had 800 patients with RNA and cell composition information, but only 300 of them had DNA methylation information. And so we had missing data and we had partial overlaps between these modes of information. So we weren't able to apply SNF or most integration approaches. So this is when we partnered with Rex Ma. Rex is a graduate student in Bo Wang's lab focused on machine learning. And he came up with a solution to encode each of these sources of data into graph neural networks that essentially look at the similarity between patients based on RNA, DNA methylation, and cell composition. And using some deep learning approaches, he's able to integrate these neural networks despite this partial overlap problem and generate a fully integrated network that uses all 3 layers of information. And so what I'm showing you here with these colorful dots is that each dot is one patient, and they're positioned based on their similarity to one another across all 3 layers of information that are available to us. And they've been colored based on subtypes that we defined based on the similarity network. Okay. Sorry. And this is a tool called Integrate Anyomics. The preprint is out online if you want to take a look. What is the biological relevance of these subtypes? Well, we had renamed the subtypes beyond the colors that you see on the left, and we looked at the genetic alterations that were driving each patient's disease. And those are the rows that I'm showing you over here. And you can see that these AML subtypes that we had developed were genetically distinct from one another. Very importantly, they differed in their survival outcomes. And they also differed in sensitivity to key drugs that are being used in the clinic today, such as venetoclax. So by taking all of this information together and using deep learning to fuse it all, we are able to generate a new stratification that is clinically relevant and biologically important. So that was really exciting for us, but we kind of ran into a challenge with trying to implement this in the clinic. And the challenge was that with RNA-seq data, with methylation data, generating this data and analyzing it can take weeks, sometimes even months, right? And AML is a really deadly disease. It's fatal within weeks if it's left untreated. So you have to make a decision really fast. And so these kind of complex analyses are not really tenable to facilitate clinical decision-making, at least for that first choice on what drug you should be giving the patient. And so we have to distill them into simpler tests that we can use in the clinic. So our lab in 2016 had previously developed a prognostic signature that uses 17 genes related to stem cell biology. And by measuring it on this chip that takes 1 day, it can predict whether a patient is going to survive for a long time or if the patient is going to rapidly decline and need more intensive therapy. So we were inspired by this approach. And what we had done was to take this existing test, these 17 genes that are already being measured, and to incorporate this new information that we had found around drug sensitivity and its relationship to the different cell types that are present within a patient's tumor. And we were able to develop a 7-gene subscore from this original 17-gene test that was able to predict drug sensitivity to 105 investigational drugs from these drug screens, and importantly, to go into clinical trials and to re-stratify patients into groups that will benefit from a certain drug that's being tested. In this case, it was a drug called Mylotarg, and groups that will not benefit. And so this is now being implemented on this single assay. It's a chip where you measure the RNA and tells you the result within 24 hours. Okay, so I'm just going to wrap off with one slide, just thinking about some key considerations that I've thought a lot about during my PhD. So, AML is a really heterogeneous cancer. It's really a group of cancers under the same umbrella. And because of all these drugs that are coming out, precision medicine is really important for AML. And I think it'll play a very important role in coming up with decisions and treatment in the future. But in order to advance precision medicine and use tools like AI, I think there are 2 parallel approaches that we need to take. So on one end, we have to use cutting-edge approaches to characterize biology really deeply, right? To use tools like single-cell analysis, multi-omics, and then to use complex algorithms like the ones I described to you to find clinically relevant insights. But in parallel, in order to implement it in the clinic, I think that it needs to be distilled. So we need to find the minimum amount of data needed for a clinical test that is affordable and that is practical. And we need to turn these complex algorithms into things that are simple. Like if this score is high, you should consider this drug. Because in the span of a 20-minute appointment, it's difficult to interpret the similarity networks of a neural network when you have to decide whether to prescribe a drug or not. So hopefully that was kind of interesting. And there's a lot of acknowledgements to have John, who has mentored me throughout my PhD, and of course Rex and Bo Wang, who were critical in helping us implement these similarity networks. Yeah, thank you so much, and I'm excited for the question period ahead.